Alright. I'm tired. So, okay, let's shut that first of all because we're going to have a limited echo otherwise. But anyway, I'm tired. Benny's tired, you could hear it. So I figured I would pop this up now. See if anybody shows up, has any questions. I'll give it a little bit and then I'll probably take a really nice nap. I was awake until like 3:30 in the morning. Just couldn't go to sleep. I'm also going to set this up because I know some people were stuck with sex-linked trait stuff. So I figured I would. Let me see. Where's this stupid thing that I need? So I figured I would transfer the sex-linked trait worksheet onto the review sheet, and then we could kind of go over it a little bit. Okay. I know people had a lot of questions about sex-linked trait stuff, so I figure that's a good as good a place as any to maybe go over some random stuff while I wait for people to maybe or maybe not show up. Stuff while I wait for people. Okay, good. Just making sure that I don't have that echo. Crud. Edit video. I gotta edit the description. Okay, good. There we go. So, a woman who is heterozygous for hemophilia marries a normal man. What will be the possible phenotype ratio of their kids? So, first of all, when it's sex-linked traits, the X is right there. Basically, remember with sex-linked traits that you have to think about whether it's X or Y. So, for example, a woman who's heterozygous. So, first thing you have to think about, it's a woman, so XX, right? Then she's heterozygous for hemophilia. We're going to use big H. Put that over here. Big H equals no hemophilia. Little h equals hemophilia because hemophilia is recessive. So once you have, okay, it's a woman, now you figure out your H's. She's heterozygous, which means mixed letters. So big H, little h. It's not superscript, but this is what it normally would look like, just like that. And then you have a male, so woman is heterozygous, marries a normal man. So it's a guy, X, Y. Okay? It's normal man, so H is normal, no hemophilia. So big H right there. Then you would do your Punnett square. And your possible phenotype ratios. And most of you, I think, were pretty good about Punnett squares, so should be alright with that. I need to go back to the video to see if anybody showed up with any questions. Doesn't look like it. A woman who is a carrier for hemophilia. Carrier means hemophilia. Jeez. Carrier means heterozygous. It means that she has the little h for hemophilia, but she also has the big H, so she shows as normal. She's carrying it hidden. And she's married as a hemophiliac man. What will their children be? So for that one, again, woman. So she's XX. Bam. So now the question is, what are her H's? Woman as a carrier, she's heterozygous, and that's what we have right here. Heterozygous. Big H, little h. And she marries a hemophiliac man. So we're going to have XY. And the question is, he's a hemophiliac, so will he have big H or little h? He's going to have little h. Once you figure that out, it's pretty easy to do the Punnett squares afterwards. And I guess just in case you're not sure, I'll do the Punnett square. Why not? So x big H, x little h, x big H, xy. Okay, so we're gonna, how do I want to do this? This way. Okay. So first of all, we get this x big H is gonna come right here. This x little h is gonna come right here. Come on. There we go. Then this x little h is going to come over here, and so is this x little h. In fact, we could even color code this. Yeah, sure, we'll leave it red and black. That works. So our x big H, our x big H. Now we got our y, our x little h, and our x little h. And then lastly, oops, I ain't gonna work the way I want it to. So males, this is the dad over here with the XY chromosome combination. And so the dad could either give X little h, or he could give the Y. He could give away one of his alleles. So phenotypes. This is a daughter who's normal, carrier. This is a daughter who's hemophiliac. She's got two little h's, one from each parent. This is a son who's hemophiliac. You got the little h from the mom and the y from the dad. And the y doesn't have anything on it. Or we could get, again, a son, XY chromosome, a son with big H. So he's normal. No hemophilia for that one. So these are, I think these are pretty easy. I don't think anybody had any problems with these. So we just did some sex-linked trait stuff. Incomplete dominance, neither trait will show up. So it's not going to be red or white. When it's heterozygous, you're not going to get red. You're not going to get white. Maybe you get pink, something in the middle. Complete dominance. This is red. This is also red, and this is white. Codominance. This is red. This is when both traits show up. So it's red and white. And then this right here is white. DNA was used. DNA that was likely copied was used because we had ourselves an mRNA sequence. And we used that mRNA sequence to figure out what amino acids to put in order. And that order of amino acids gives us our protein, represented as our final traits. So that's how all of these things were represented in the Snork lab. Their products were all used or shown or represented in some way, shape, or form. Why did we flip the coin? Whenever you go to have a child, for most traits, you're going to pass, one allele, one letter, like a big T or a little t, to your child for a certain trait. So, like T is red and little t is white. So let's say you happen to be a mix. Let's say you happen to be big T, little t. You have a 50-50 chance. You could pass that, big T, or you could pass that, little t. So a coin flip is a 50-50 chance. So that's why it represents our way of passing alleles to our children. If you have two big T's, well, which T are you going to pass? You got a 50% chance of that one, or a 50% chance of this one. In that regard, it doesn't matter quite as much, but that's just how it works. The parents were all heterozygous, which means none of them had a recessive trait. So that's one reason the kids, because of the 50-50 chance, they could end up inheriting a recessive trait because they could get both of the recessive alleles from their heterozygous parents. Also, there were certain traits that were incompletely dominant, which means that the heterozygous is a unique trait, or variation, I should say. So if the kids were pure dominant or pure recessive for those particular traits, then they would have a very different phenotype from their parents. So multiple reasons for this question. So on the test I might ask like, hey, two different reasons, because there are two important different reasons. Alright. I think everybody was pretty good with this stuff. Remember, AUG is the start codon. You do not start making amino acids or tRNA until you see that. If you don't see that, you don't start. And then stop codon means stop. You don't continue any further after that. So, you could slightly heat the what the heck's it called? The test tubes. If it's room temperature, maybe you heat it to closer to body temperature, since the enzymes that are needed would probably work best at body temperature, since they're usually inside your body. Or maybe you just add more enzymes, so that maybe they'll bind to substrates more often because there's more enzymes. Or you could change the pH conditions to come closer to what would be the pH conditions inside of a nucleus or a cell or whatever. So there's a couple different potential options there. Alright, so I think we're pretty good. No one showed up. 16 solid minutes almost of some review. So hopefully if someone pops up on this channel later, they'll see some good review stuff. And ask a question, and hopefully later, when I am more conscious hopefully I'll see it and I'll be able to come back and answer it. All right. Peace out, everybody.